In this week's lecture, we're going to be covering one of the two mechanoreceptors, mechanosenses. And the reason why they're called mechanosenses is because they require the pushing or a physical depression of the receptors by the stimulus in the various sense organs. So in today's lecture, we're going to be talking about hearing, which is also known as audition. You might be wondering, how in the world can sound waves actually push and pull? Well, we're going to talk about the way in which sound ends up getting converted into movement and fluid, which ultimately causes the movement of the receptors, allowing the transduction of sound. So sound is also known as audition, or hearing is also known as audition. So the kind of stimulus that we detect with our nervous systems is known as the auditory stimulus. So sound waves or auditory stimuli are actually just sound waves or air that has been either compressed by the sound waves or rarefied, which is separated. So these changes in air pressure that you're seeing here represent the, downward and upward and downward and upward appearance of sound waves. All they do is represent compressed and rarefied airways. And this causes, as the sound enters our ear into the ear canal, this causes our eardrum to vibrate in response to that compressed and rarefied air. So here, just to illustrate to you how the compressed and rarefied air matches with the sine waves of the sound waves, it's shown here. The compressed air represents the peak in the sound wave, whereas the rarefied air represents the trough. But essentially, changes in air pressure is what gives rise to sound waves. There are dimensions of sound waves. So amplitude, which is the height of the peak, represents how loud or soft something is, right? We perceive it as loud and soft, but it's the amplitude of the sound. The frequency, how fast we have a cycle of the sine wave, represents the pitch. The faster the frequency, the higher the pitch. The slower the frequency, the lower the pitch. Most sounds we hear are not in their cleanest form. So most sound we hear has some element of complexity to it, and we perceive it as timbre. So most sound is not simple to us, although it can be. So for example, my voice is distorting the sound waves in your ear, but my voice is not simple. It has a complexity that allows you to distinguish my voice from anybody else's that you hear that might have the same pitch or same amplitude. But the complexity of the sound is what allows us to determine the timbre of the sound waves. Right? Being able to distinguish between a trumpet playing the same note at the same volume from an oboe or a piano playing the same note at the same volume. So let's talk about what in our ear allows us to actually detect sound. So don't be overwhelmed by this diagram from your textbook, but we're going to go through it many, many times. But the ear, or the hearing system, can be broken down into three divisions: the outer ear, the middle ear, and the inner ear. The outer ear is what you usually see. It's your earlobe. Also, a fancy word for earlobe is pinna. And sound enters through the pinna. It's funneled by the pinna into the ear canal. So here's the ear canal. And then as sound waves get compressed and rarefied, it causes the tympanic membrane or eardrum, which is the last portion of the outer ear, to vibrate back and forth, right? It moves back and forth. The vibration of the eardrum causes the middle ear, which has these super little tiny bones called the ossicles, to vibrate back and forth as well. So as the eardrum vibrates back and forth, it causes movement, physical movement, in the ossicles. The ossicles, the last of the three ossicles, are laying against a very thin membranous structure of the inner ear called the oval window. So what it's doing is it's kind of pushing the oval window. As the ossicles move back and forth, this causes movement in the oval window to occur, which ultimately moves fluid in the snail-like structure that we will focus on in this lecture called the cochlea. So in your inner ear, you also have a portion of the cochlea that the inner ear that we're not going to talk about that's important for balance. But we're going to be focusing on the cochlea, which is important for transduction of sound. So I just threw a ton at you with this image, but here is a more, described, simplified version of it. So as I mentioned, there are three divisions of the ear that are responsible for helping us transduce sound. We've got the outer, middle, and inner ear. The outer ear consists of the earlobe, the pinna, the ear canal, and the tympanic membrane or eardrum. The function of the outer ear is to funnel sound into the middle ear. The middle ear contains those little bones called the ossicles, and a portion of the inner ear called the oval window against which the ossicles rest. Their function is to amplify sound. So as the outer ear causes them to vibrate in response to the sound waves, they vibrate even more and ultimately transfer this movement to the inner ear. The inner ear is made up of the cochlea, that snail-like structure that I showed you in the previous figure, and its sole purpose is to detect or transduce sound. It takes these vibrations from the middle ear, from the ossicles that it is transferred to, and converts sound into a neural impulse. So let's talk about the way in which this happens. So just to kind of illustrate this a little bit differently from a different figure, right? We've got the tympanic membrane here that vibrates back and forth, and we've got the bones of the middle ear resting up against the oval window. So as the bones of the middle ear move, they cause the oval window, which is very thin, to move back and forth. As it's moving back and forth, it pushes the fluid that's filling the cochlea back and forth as well. So don't worry about any of this stuff just yet. We're not going to even actually worry about it. But I just wanted to illustrate to you a different way to look at the way in which physical vibrations in the bones ultimately causes movement of fluid in the cochlea. So we've got an area of the cochlea called the round window. So as the middle ear, the bones of the middle ear push the oval window, right, fluid in the oval, against the oval window, the fluid has to compensate, right? This is a closed system, so the fluid has to have some sort of way to move out as the oval window is being pushed in. And so the round window serves as the opportunity for the fluid to kind of take away that pressure so our inner ear doesn't explode when the fluid is pushed by the middle ear bones. So this image is from your textbook. We've blown up the cochlea here, shown in red, down here below. And so what you're actually looking at is the cochlea has been sliced, and you see that there are actually many compartments that make up the cochlea, this winding, twisted, snail-like structure. Within the cochlea, each chamber has been blown up over here, there's actually a specific organ that transduces the sound, and it's called the organ of Corti. So that's shown over here, right? The organ of Corti is shown here, and what's responsible for actually transducing sound are hair cells. So we'll talk about how they do it in a second, but I want to call your attention to what seems to be cut off here is the basilar membrane. So the basilar membrane sits on the base of this organ of Corti, What happens when fluid moves past it is that the basilar membrane flexes up and down. The flexing of the basilar membrane causes the hair on these hair cells to move back and forth, right? Think about when you lay down in a bathtub filled with water or in a pool, your hair kind of goes along with the flow of the water that it's in. These hair cells are no different. When fluid moves across them, they move back and forth, okay? And these hair cells are the ones that are actually responsible for transducing sound, and you'll notice that the auditory nerve, which carries the information to the brain, is connected to them. So let's take a look at the basilar membrane in a little bit more detail. Actually, first we'll talk about the organ of Corti again. The organ of Corti, right? So if you slice the cochlea, you'll see that each chamber has 3 areas, 1, 2, and 3. And in this middle chamber, we have the organ that's actually responsible for converting sound into an electrical impulse. And so here's the blown up version of it. We've got hair cells that are called the outer hair cells that are embedded in this roof-like structure called the tectorial membrane. And we've got inner hair cells whose hair cells are kind of floating free in the fluid. The hair cells, the inner hair cells, are responsible for transducing sound, and it's detected by the auditory nerve and then carried to the brain. A fancy word for the hair that can be found on the hair cells is stereocilia. So don't worry about these other terms that are shown here, but I like this figure from a different textbook because it very nicely kind of blows up this image of what the organ of Corti looks like than your textbook actually did. Okay, now let's talk about the function of the basilar membrane. So the basilar membrane, just to kind of illustrate to you, is right here. And in this blown up image, it's over here. So the basilar membrane serves as like the foundation for all these cells that are hanging out, waiting for the fluid to move past them. So remember, as the oval window is pushed by the ossicles, it's going to cause fluid to move back and forth across these chambers. Ultimately, it causes the flexing of the basilar membrane to move back and forth. And this flexing is really important because where the basilar membrane flexes is where pitch or frequency is registered. So take a look here, right? So this is the cochlea here. It's actually a wound up structure. And if we unwind it, this is what it would look like, the uncoiled cochlea. And here's the basilar membrane right here. Okay? The basilar membrane has a base, the area located closest to the oval and round window, and it's also got an apex that's kind of, deep within the cochlea when it is wound up. So different parts of the basilar membrane flex and register sound frequency or pitch. So the basilar membrane is going to move up and down as fluid moves past it. Remember, this whole thing is filled with fluid. And so as the fluid moves past it as a result of the ossicles pushing against the oval window, it's going to cause the basilar membrane to flex back and forth, but only flex back and forth in specific locations. The frequency of the sound is registered differently. So higher pitch sounds, are going to flex the basilar membrane closer to the oval window, whereas deeper sounds, or lower pitch, are going to cause the basilar membrane to flex further away or at the apex, right? So high frequency sounds, as you can see here, cause the basilar membrane to flex closer to the oval window, which is right here, whereas low frequency sounds, or low pitch, cause the basilar membrane to flex closer to the apex. So where in the basilar membrane the sound causes it to flex allows us to perceive different pitches of sound. So remember, we've got hair cells embedded in each of these 2 locations, right? So when we hear high pitch sound, it causes the hair cells to move here at the base, but not at the apex. So our brain translates the location of the flexing of the hair cells at the base into higher pitch sounds. Right? This is pretty incredible, but this is exactly how sound is perceived as pitch, how sound waves get converted into an electrical impulse. So if you want to take a look at this video, it kind of unravels, allows you to walk into the ear and inner ear and unravels the cochlea, but then it plays a song that you should recognize, causing the membrane to flex in different ways as we listen to music. So just to recap and to remind you, the inner hair cells, the movement of the hair across the fluid as the basilar membrane flexes, causes the sound waves to be transduced into an electrical impulse. If you'd like to know how this happens, I'm happy to share this with you, but you don't need to know. You're not required to know how transduction happens in the ear. But let's talk about how this information gets from the organ of Corti, which resides in the inner ear, to the brain. So the auditory pathway, right, starts at the organ of Corti, where the inner hair cells transduce sound. This information, right, these inner hair cells cause the release of neurotransmitters, which are detected by the auditory nerve, these axons of the auditory nerve, and the nerve bundles together and heads to the brain. Where does it go? So again, don't be overwhelmed by this, but this is an image from your textbook. So it's as though you were looking at a person head on and you're seeing a slice, a frontal or coronal slice through their brain. So information, here's the inner ear. Look how tiny it is in your actual face, right? So the inner ear has inner hair cells that transduce sound. Sound information travels through the auditory nerve, and the first place it synapses is the cochlear nucleus. And it's called the cochlear nuclei because there are two on either side, shown here in red. It's part of the medulla. From here, it synapses with the superior olivary nucleus. We're still in the medulla. What's important about these two nuclei, which we won't be spending time on, but we'll be discussing it in a second, is it allows us to detect location of sound. So you can close your eyes and know the direction that the sound comes from, largely because of the arrival of sound being different from your left and right ear. And what allows us to detect that is the superior olivary nucleus. So from here, information travels to the inferior colliculus, which is found in the midbrain. The inferior colliculus allows us to have reflexes to sound. So if we hear a sound, it forces us to kind of turn our head and attention into the direction of sound that it came from. And then it finally, after what seems like forever, synapses with a nucleus in the thalamus called the medial geniculate nucleus. The medial geniculate nucleus is the relay nucleus for the auditory system. And then from here, it finally goes to the primary auditory cortex, where perception actually occurs. So we perceive sound for the first time when it hits the primary auditory cortex, but not prior to that. Incredibly, the primary auditory cortex has a tonotopic map of the basilar membrane. So what that means is, remember, the base registered more high frequency sound, and then it gradually registered different frequencies of sound, where the apex handled, more deeper sounds. So you have a representation of that basilar membrane from low to high pitch in the primary auditory cortex, and here it is. So this is the sagittal view of the brain. The nose would be right here. The back of the head is here. Here's the top of the head. This is the temporal lobe shown here. right? And within the temporal lobe is the primary auditory cortex. And if we blow up the primary auditory cortex, you will see that different areas of the primary auditory cortex are responsible for perceiving or processing low to high-pitched sounds in the order of low to high, right? So there's a physical representation of the basilar membrane in our primary auditory cortex, which is pretty incredible. So how do we perceive sound location? I just mentioned to you that there are areas in our brain, such as the superior olivary nucleus in the medulla, that's responsible for detecting arrival of sound. And that's one of the ways that we are able to detect spatial location of sound. We tell the location of left and right based on the difference in timing and loudness of the sound that arrives in each ear. This is known as the duplex theory of sound localization, and this is the way it works. When we are walking and we hear sound coming from a particular direction, it's going to hit our right ear first before it hits our left ear. So that's the difference in timing. Additionally, I'll show you in the next slide. So sound waves, depending on the direction that they're coming from, are going to hit our left and right ear differently. And what you're seeing here is the time delay between each ear. So when sound waves are coming from, let's say, 1 o'clock or 2 o'clock, it's going to hit our right ear first, and after about 0.3 milliseconds, it's going to hit our left ear. But incredibly, our nervous system is able to pick up such a quick difference in sound arrival and allow us to determine the location of sound. The other way in which sound location is determined is the intensity of sound. So as the sound source hits our head, right, it's going to sound louder to our right ear than it will to our left ear, because our head creates a shadow for these sound waves, and they don't exactly, hit our left ear head on. And so this difference in intensity of sound, as well as the timing of sound hitting our two ears, makes us perceive the location of sound. And so again, just to kind of illustrate this difference in intensity of sound, so sound waves that are coming from our right are going to sound louder to our right than our left. Sound waves that are coming kind of at an angle around 1 o'clock is still going to create a shadow to our left ear. Sound waves coming head on sound exactly the same loudness from our left and our right ears. And again, these differences in sound intensity as well as arrival of time of sound allows our brain to perceive location of sound. Okay, so this just kind of summarizes the left and right perception of sound direction is a result of the duplex theory of sound localization, right? Difference in timing as well as loudness. But for front and back, right? Front and back, sound arrives at the same time and sounds the same intensity. And so what contributes to our perception of sound localization is the distortion by the pinna. You'll probably notice that your ears don't have the same bends and twists in your earlobe on either side. This contributes to a difference in timbre, right? The complexity of the sound, and our brain perceives this as difference in location, whether it's front or back or top or bottom. So just to illustrate how the pinna contributes to the difference in perception of sound localization, sound enters and hits our pinna in different angles, and this enters our ear differently in our left and right ear, allowing us to perceive sound from the front or the back. Okay, so let's summarize this shorter lecture than usual. Hearing is the result of changes in air pressure, right? The compressed and rarefied ear that ends up vibrating our eardrum or tympanic membrane. This vibration is amplified by the bones of the middle ear, the super tiny bones known as the ossicles. Sounds are transduced, I should say are, transduced by the inner ear, specifically the cochlea, as the bones push up against the fluid of the cochlea against the oval window. Depending on the pitch of the sound, different areas of the basilar membrane found in the cochlea flex. The higher pitch sounds cause the basilar membrane at the base to flex. The lower pitch sounds cause the basilar membrane at the apex to flex. The organ of Corti in that portion of the basilar membrane is what transduces the movement of fluid into the electrical impulse which is carried to the brain. And the cells that are actually responsible for the transduction are the inner hair cells, whose hair kind of moves with the fluid as needed. They release neurotransmitters onto the dendrites of the auditory nerve, which carries sound info to the brain through the auditory pathway. The brain has a tonotopic representation of the basilar membrane, and lastly, sound localization is determined by the duplex theory of sound localization, as well as the distortions produced by the pinna. So that's it for this lecture. Don't forget to submit your reflections for it.